Can someone confirm that you can hear me?
We can hear you.
Thank you. Sorry for being late. I forgot that I have to log into Zoom to start my well, the Zoom. Um, I was trying to get in through clark.edu and it just isn't working. I'm not sure what the problem is. Um, all right. Are there any questions before we begin? Nope, I'm in the lecture. That's not right. All right, before we begin, let me state that I've done some grading. I have graded, oh, what the heck is it? The study credit for week 2 and week 3. I've graded the last two lecture worksheets. I think it was lecture worksheet 3 and 4. And I've graded one of the labs. I think it was lab 4. Any questions about the grading?
Are you going to be putting in that extra credit study sheet soon?
I haven't graded the extra credit study sheet. So I'm not sure when I'll get it graded, but I haven't graded it yet. Sorry about that. Um, Come on. There we go. All right. I don't have the grade book up anymore, so I'm gonna share the screen. Might as well optimize video sharing. There you go. Should be seeing my screen now. So today is October 21st, and we should be discussing the unknown project today. Let me remind you, there's going to be a quiz this week, quiz 2, covering labs 3 and 4, and chapter 3, and the end of chapter 2. Any questions about any of that? All right, let me go to the unknown project. Where do I have that? I think it's not in the lab, but let me look. I'm going to use this just because it's easier if I display it, and it displays better than if I run it in Canvas. But you should have access to it in Canvas, and that looks like the latest one. All right, the unknown project is a project worth 75 points. It is a lab project. It's following the time-honored part of microbiology courses, although you're not actually going to be doing this project because everything will be done virtually, meaning online. I will give you a number which represents a bacteria species. Let me go ahead and show you the species. So I'll send you a number, and one number will represent each species. And so here we have the gram-negative bacilli, and there's about 20 of them. And then we also have some gram-positive cocci, and also there's about 20 of them. So one number will represent one of these species, and then um, all term long, you'll be requesting tests to try and identify the species. And here are the tests that you can request. Up on the top, there. Another place to see the tests is to come to get out of there. Actually, I do need to keep up that one. All right. Another place to see the tests are to come here under week 5 lab. And there's week 5 lab unknown tests, and this is all the tests that you can request. All right, any questions about that part of the project?
So would we just email you about the tests that we want or want to request?
No, you don't email me the tests except for the very first one. I'll be emailing you 3 tests. You are going to use your unknown notes pages for requesting tests. And let me stop the share because I need to call up an unknown notes page from an earlier term because nobody's obviously submitted anything this term. But let me, and I got to pull off the grade so you guys can see it. That's why I need to do this. I'm in the wrong place here. Let's go back to. There we go. Let's call up dashboard. Trying to get there. Just give me a minute. Grade, and then I can show you. Share my screen. All right. So this student has requested actually several tests, November 12th. Let's go to that date. This is the first time the student did it. And what they did was they did the gram stain on October 22nd, the gelatin hydrolysis tame on October 23rd, and the PR lactose test on October 24th, and then got the results to all 3 of the tests on October 29th. Let me blow this up a little. And there's the gram positive, the gram stain results. And October 31st, I don't know why they have a different date there. They have the gelatin hydrolysis test. And November 21st, the PR lactose test. And then gave the results there. And then the next test they requested was the hemolysis test, which they requested on November 12th, it looks like. And you can see the student has requested different tests on different days. They put in a test request on November 19th. I'm just going to scroll down to it now. There's the hemolysis test result. And the next test they requested was the 6.5% sodium chloride broth test. Any question about how you request tests?
So do we request them and then you just tell us the results?
Um, no, you request a test and then I'll send you a picture of your results. You have to put in the results and you put in your interpretation. Like right here, the student requested the hemolysis test above, meaning not on the state. Where is it? November 19th is when the student put in the results, but I sent it to them November 16th. I sent the results, and then they put in the picture of the results and they gave the test request that they requested, and it looks like there's no change in the hemolysis test, and so they concluded that the conclusion is gamma hemolysis. Now the results is the what do you call it? The observation of the test. That's your result. And then the conclusion is your interpretation of the results. So gamma hemolysis is the interpretation of the result, and the result is no change in the blood auger. So that would be the result or the observation. Any question about any of that? So when you put in your results, you should put in the picture I showed and then give your observation. That's the result. And give your interpretation of that observation. And that would be the conclusion. So you should put in both the results and the conclusion. Anyways, you request a test in the unknown notes page, and this is showing you an unknown notes page for a student. And I will send you in your unknown notes page the result, and I'll send it in the comment section. Students should not use the comment section. You should put your comments, if you have a comment, in your document and then submit the document. You can submit an infinite number of documents in the unknown notes pages. And you don't have to worry, everything will be dated. Actually, it's even timed. And then I will submit it in the comments. If you start using the comments, I'll actually start deleting the comments, and I'll warn you, you should put that in your unknown notes submission because I'm going to delete it. So don't use the comments. I use the comments. Any question about how you work with the unknown notes page? And how you get the unknown notes page is scroll down to week 5 lab. There's the instructions I'm talking about now. And the unknown notes page is right there. That's your unknown notes page. I can access it and grade everybody's, but you will only have access to your own unknown notes page. And instead of having what is that called? The speed grader, you'll have either a submission button or a download button or an upload button, something like that.
And the only place we can find the unknown notes is week 5. That's the only place we can find it.
You can also find it from the grade book, but this is the best place to find it, week 5 lab.
Okay.
And there's the instructions for what you need to put in the unknown notes page. I haven't talked about that. Let me blow this page up so you can read that. After the first 1 or 2 emails from me, please record all of your results and interpretations, meaning your observation and conclusions in your unknown notes pages here. Then record your daily activities, meaning what tests you're going to request here. You should put the test request either at the end of the document, that's where I ask you to put it, but if you have it at the start of the document, I'll see it. If you put it in the middle of the document, I will not see your test request until I read the the unknown notes document you submitted. And every day, or almost every day, certainly every other day, I will be looking at the unknown notes pages, but I'll only be scanning the end of the document. And then, of course, when I open it up, I'll see the beginning of the document. So if you put your test request someplace in the middle of your unknown notes page, meaning your document, I will not see it when I look for the test requests. And you'll have to wait until I actually go reading through your unknown notes document before I'll find your test request. So put it at the end of the document, and then I'll find it. You should also put everything you do in your unknown project in your unknown notes page. The reason why I'm doing this is I want all of the data to this project to be put in one place and easy to find for both you and for me. So we can go look up what you've done in your unknown notes pages for the unknown project. The system will date everything when you submit it. It'll be dated also when I submit something in the comments. Please don't use the comment section. Put everything in your submission pages to make the document. All right, any questions about how you use the unknown notes pages? And then one other thing is that when you request a test, you should be following dichotomous logic. And let's go ahead and get out of here now. Oop, I need to put this. I need to stop the share and then put the student's grade back. This isn't an early grade, but in case the student wants to look up what they got here.
I have a question.
Just a minute, let me put the student's grade back in their unknown notes pages, and then I will put that back and then close this down and then open up the share my screen. All right, what's your question?
I don't know if you remember, you sent me, I requested the results for the test, and you sent me the results, but I can only see halfway. I can't see the whole page.
Were you in a class of mine in an earlier term? Because I haven't sent out any results for this term.
No, you did. I request for the plug quiz,
The what? To quiz?
the plug.
Plagy quiz.
Yeah,
Okay,
that one.
that's not for the unknown project. I haven't
I
sent
mean,
out any results for the unknown project.
now they are not the test. So I did request, and you sent me, but I can see it. It only shows halfway. It's not showing the full, like the whole thing. Like I
Um,
can see the whole screen. It's only showing half of the screen. So
let
I don't
me, I should probably deal with this later, but let me just look up your unknown notes page just a minute. Let me stop sharing the screen. And
I mean, I don't know if it's gonna show on unknown because I remember when I requested it, we were here on Zoom class and it was the end of the class and you went and checked my profile and you sent me what I needed.
I sent
So I
it
don't,
to you in email, is what you're telling me.
you sent me via, yeah, the Canvas email.
All right, let me take a look at the Canvas email and who's talking? I'm not seeing that because I'm not sharing my screen.
Oh,
So
it's
who's
me,
talking? Uh-huh.
Chancellor Madeline.
All right. Like I said, I haven't sent out any unknown notes pages, so or any unknown project results. So I'm not sure what you're talking about, but let me look up your email. I'm trying to do a search here now. Oh, come on. Come on. Found you. All right. Surprising, there's a whole bunch of chancellors in my class. A request. I see a request for what you missed on the Plagy quiz. That's not the unknown project. You also turned in a bacterial identification, microscopic techniques. This looks like it's a student group report for Microbiology 260. And what did you request for the very first time? Oh, you asked about some quiz results. I don't know if you ever sent it to me. And I asked you, what quiz are you talking about? So maybe it was the environmental quiz. It's another email. So you haven't requested any unknown notes or unknown project tests, Chancellor. Okay. Maybe you're requesting
Well, I don't
Madeline
understand. You
document.
sent me something.
Yeah, I
You sent
sent you a document called Madeline document. You should be able to open the file in Microsoft Word. And then I'm showing you the questions you missed in the plagiarism quiz.
So that's what I'm talking about. The one that you sent me, those questions, I can't see. I
Do you have
can't see it
Microsoft Word
clearly.
on your
I have
computer?
Mac.
I'm sorry?
I have a MacBook.
Okay, do you have Microsoft Word?
Yeah, I do.
Okay, so you download the document, then open Microsoft Word, and then open the document in Microsoft Word.
Let me see.
All right, that's
Okay.
enough. That's not the unknown project. I'm getting out of here and I'm coming back to discussing the unknown project. Let me share my screen. All right, what did we mention? It's 75-point lab project. And how you're going to be doing this project, you'll be using your unknown notes pages to request the tests. The exception will be the first 3 tests I'm going to send to you in an email. And the reason for my doing this is this project will have about 5 to 6 student requesting tests for this project. And I've got about 40 students now. You do the math. 40 times, let's go 6. I don't even know how much that is, but I'll get it here. There's going to be 240 student interactions with me. And 240 student interactions is just way much for a project. And so I talked to the other microbiology instructors. How about if I send them 2 or 3 of the first tests, and I'll send the whole class one email instead of, let's see, 40 times 3. And I have to go and equal at the start of this. So that will reduce the number of requests by 120, meaning on average, there will only be 120 requests for this project. That's actually an awful lot of requests for one project, but it's a number I can deal with. So I'm going to send you the first 3 test requests. I've just decided what they will be, and you will not request the first 3 test requests. And instead of sending out 120, I'm just going to send out 1 email. And that reduced the number of interactions on this project with my students by about 120, making it about 120 test requests that you guys will be making. That's doable. Okay. Any questions about any of that? So the first 3 test requests I will send to you in an email. And I've decided, can you see the page here? I've got the bacteria here and the test request there. I'm going to send you the gram stain because that's a real good test request to have. It separates the bacteria into 2 large groups. Oops, sorry about that. I'm also going to send you the gelatin test because this test takes a week before you can get the results. So if you request the test today, which is what I'm going to pretend you're requesting, you won't get the results for 1 week. It's the longest test we have. And then I'm also going to send the PR lactose test just because it's a good test to run for at least most students. Some students, it doesn't give much results, but like I said, it's a good test for about most of the students, about half the students. Any question about that? So the very first test, I've decided which test you're going to be running, and it's the gram stain, the gelatin hydrolysis, and the PR lactose. This will be the only test you will get the results in the email. And I'll just send out 1 email to the whole class. And in that email, you also get your unknown number representing a bacteria species, and then the results for these 3 tests. You should take the number and put it in your unknown notes page, because when you request a test, if I don't have a number, I can't send you the results to the species. So you must put in your unknown number in your unknown notes page. And then you should put the results and the conclusions for the gram stain. The result would be the cell is either purple or pink, and then the interpretation is it's gram negative or gram positive. And then put in the observation on the gelatin hydrolysis test and the interpretation, and then put in the observation on the PR lactose test and the interpretation, meaning what its conclusion is. Any question about any of that? So put all of this in your unknown notes pages, and then at the end of the document, request the next test. For example, let's say you have a gram negative, and it's gelatin hydrolysis negative. And the PR lactose conclusion is it's AG. We'll talk about that. AG stands for acid and gas. You can find an interpretation of the test down here, except AG isn't here. A stands for acid, G for gas, and you have either negative, meaning there's no change in the tube. You have A for acid, AG for acid and gas, and then there's a V down there, and that's in here. V stands for result is variable. Let me show you what each of these looks like. Make sure I have that highlighted. Yes, I do. Good. All right. All right. You'll notice there is no PR lactose, as well as any of the others. Where I have that. There it is. There's no other PR glucose, PR lactose, PR sucrose, PR mannitol, PR inulin. What we call it in the test is PR carbohydrates. And that's all of the above. Let me go ahead and open that. Because the PR carbohydrates is actually PR glucose, PR lactose, PR sucrose, PR mannitol, PR inulin, or another carbohydrate. And what you do is you have the PR broth, and then one of the carbohydrates is added, like glucose for the PR glucose test, lactose for the PR lactose test. So a single fermentable sugar carbohydrate is in the tube, as well as the phenol red, that's the pH indicator standing for PR. And then there's an inverted Durham tube in the broth. Let me show you the picture of that. There you go. So there's a tube that's PR negative, and there's the inverted Durham tube, and it's no change. So the color is either pinkish or reddish. Get that out of there. And this one is going to be PR positive because it's turned yellow. And you can put that as A for acid. Acid makes the pH indicator turn yellow. And then it's also got gas in it. The gas is collected by the inverted Durham tube. And so if you see a bubble in that inverted Durham tube, it's gas. You only see gas if there's an acid, so it'd always be Ag if you have gas, or A if there's no gas. Oh, come on, why am I not getting up on this page? And then there's the methods for doing the tube. You get 2 PR carbohydrate broth tubes. Inoculate one with the bacteria. Keep the other uninoculated. It is your negative control. You incubate them at appropriate temperature, whatever temperature your organism grows well at, and incubate it for 18 to 48 hours. Beyond 48 hours, it's too long, and the tube should not be used, so it should be incubated for 18 to 48 hours. Take it out of the incubator. Note that there's growth. You take a look at the color of the tube and whether there's gas formation. If it's yellow, you interpret the conclusion as acid, and if it's yellow and gas, you write the conclusion as Ag, acid and gas. And if it's negative, you write negative for the tube. Any question about any of that? So when you request a test, you should read the test in the week 5 lab unknown test, and you should take a look at how long that test incubates. The PR carbohydrate broth test incubates for 18 to 48 hours, and so you will not get your results once you request the test for at least 18 hours to 48 hours. And also, I may not be checking the unknown notes pages every day. I will check them about every other day for sure. And oftentimes I'll check it every day of the week, but I will not check them at all on Sunday. And so if you request it on Friday, you may not get anything until Monday because I may not request it. If you request it on Friday after I've already looked at the unknown notes pages, I won't look at it again until I might look at it on Saturday, but I may not look at it until Monday, and I will not be looking at it for sure on Sunday. So my point is, if you request it on Friday, and I've already looked at the unknown notes page, you may not get any results until next Monday. So If you request it, you will not get any results until at least 18 to 48 hours because the test has to incubate. And I will start the test once you request it. And of course, obviously, I read it. So you should look up when the test will come because if the test, most of them take 2 days, you're not going to get the results for at least 2 days after you request the test. And it may be longer. Like I said, if you request the test on Friday, I may not give you the results until Monday. Any question about any of that?
So are we requesting, how many do we request the tests?
This project is following dichotomous logic. I started talking about that and I never got back to it. My apologies. It's in the, where is that? It's in the project that this project is following dichotomous logic. That means you can only request one test at a time. And even when I'm sending you the first three, you're following dichotomous logic because you're going to start the gelatin hydrolysis test today. And you will be starting the PR lactose test next Monday. And you will be doing the gram stain next Tuesday. And then I'm going to send you all the results to all three of the tests next Tuesday. And so you will be following dichotomous logic, even in the first three. Any question about any of that? So the test should be using dichotomous logic. If you don't use dichotomous logic, you will get a penalty. Shotgun logic is where you take a shotgun and you just run a whole bunch of tests at one time. You have a substantial loss of number of points if you use shotgun logic where you just request all the tests. Some students do that. They wait until the very end and then they just request a whole bunch of tests. If you do that, you're following shotgun logic and you will be getting a severe penalty for following shotgun logic. If you request two tests at one time, you're not following dichotomous logic, you will get a penalty. I don't remember what it is. Let me go look up on the grading here. I don't have it here. Let me look up on the grading here.
How do we know which test to request, or do we just request all of them at some point?
No, you request one project at a time.
I know it's one at a time, but I mean, like throughout the whole term, should we be expected to request every test available or only a few?
Only what you need. So you should be requesting total of something like 5 or 6. Maybe if you're unlucky, 7. But where do I have the grading here? Oh, here it is. This is going to be hard to read, but if I blow it up, you can read it. Dichotomous logic. All tests that were performed were relevant. So if all tests were relevant and you follow dichotomous logic, you'll get, let me check the adding of this. Yeah, this is out of 100, not out of a, this project's worth 75 points. So that isn't quite right, the grading sheet I'm showing you here. Someplace I've got the grading. Let me look up. I must have it online, which is why don't I have it? Is a good question. There it is. And I should download it so I get it. I don't see why that, but so if you follow dichotomous logic and all the tests were good, then they have no excess tests, so you get 10 out of 10. You have one excess test, 7 out of 10. Two excess tests, you get 5 out of 10. Shotgun logic, 3 out of 10. And then no logic at all would be 0 out of 10. Very rarely have I gotten a student who had that. Let me download this so I get access to it. Come on. Why can't I come to the top of the page here? Zoom and Canvas are not working correctly. If you did not notice, Canvas was totally down. I think it was Sunday, and it was actually down all over the United States. Let me download this, grab that, and I'm going to move it to my unknown project now. Come on. Well, I didn't see it. There's one here. I just don't see it. Is it that one? No, it's that one. All right. All right, it was there. I just didn't find it. All right, so the project format you're going to receive a pretend culture, a virtual culture containing a bacterial species. Some species will be gram positives, others will be gram negative. Virtually, you're going to isolate each species into a pure culture. So you pretend to do that, you streak it out, and you're doing that kind of now. And just write it up in your unknown notes page that you do it in your unknown project. Just write it up that you're virtually isolating the species. You need to do that 2 times. Send the cells through 2 colony isolation events to make sure you have a pure culture. And then identify your species. You can use the tests that you'd like you can find Berge's manual online. There's one in the lab. If you happen to go into the microbiology lab, you can get it from the library as well. You can use the textbook, you can use the web, but if you do use the web, make sure your results are correct. So go to a good site and then verify that the results at another site, meaning if you use the web, you could get incorrect results, incorrect interpretation, because anyone puts up anything on the internet. So if you use the web, verify that it's correct. And then get your results and make your conclusions, your interpretation of the results, write it up in your unknown notes page, and then request the next test using dichotomous logic. And how you do that is your daily unknown notes page. You should write all of your lab notes in your unknown notes pages. If you write your unknown notes page formally, meaning with formal writing, formal wording, and you put all of your unknown notes together. Let me see if I can go back to that. Nope, I don't have it open anymore. If you put everything together in your unknown notes page, and what you do is you just have your test 1 at the top of the page, test 2 down the page, and test 3, the last test at the end of the page, you could just copy your unknown notes and then paste it into your unknown report. So that's one advantage of putting everything together on your unknown notes pages, but you may put a separate document for unknown test 1, unknown test 2, and unknown test 3, and you'll just have 3 different documents. And you have to copy, if you're using formal writing, you can just copy from all of your documents and put it in your unknown report. You don't have to use formal writing. You can use informal writing in your unknown notes pages, but your unknown report should be informal writing. Any question about any of that? And what you do is you just take your data from your unknown notes page and then rewrite it up in formal writing for your unknown report.
So after the 3 tests that you send us, once we complete those, that's when we start requesting tests?
Yes. All right, your unknown report will be worth 75 points. It should be written using bullet points. That's not terribly important. You can use text. You can use an essay style. I should change that. Please use 12-point font. I wear reading glasses for a reason. If you have less than 12-point font, I'll have a hard time reading it, and I'll be a little more less lenient on the grading. So use 12-point font. Do not double space, so use single space. You should, in your unknown report, write your daily descriptions in the test you run, the information you hope to gain from these tests, the observations you made from these tests, and then the interpretations or the conclusion of that test. You should not omit data that makes your project look better. Drawings, photos, and tables are very helpful. They are not required, although you must have one table or drawing. And in your unknown report, you should put the date of the entry, meaning if you requested the test on, I don't know, October 29th, you should state that, and then you'll get the results likely on a different day. Very few of the tests would you get it on the same day, but and that would be only if you request it, and then I look at the unknown notes page on that same day. You could possibly get it on the same day, but most tests you will not, because most tests take 2 days to run. There are a few where you could get it on the same day. And then put the date of the test results. Most of the tests, the request will be on a different date than when you get the results. You do not need to put in the step-by-step details for the test runs. All you need to say is something like, I did the gram stain on October, let's see, it'd be 6 days, 27th or 28th, something like that. And then give me the gram stain results and the gram stain conclusion. The cells came out purple round cells, and the interpretation is it's gram-positive cocci. Thoughts and words in your report should be using your own words. If you use advice from another, like the instructor, it should be put in quotes, and you should give a reference. It is mandatory that you have a test table or a diagram giving description, outlining the process you use to identify your unknown organism. An example suitable table is provided below. Let's go down to it. Let me shrink that down a little so it's a little better on the page. Why aren't I seeing the very left part of this page? Can't get it. So the first test this person ran was the gram stain. And the result was, and actually the conclusion, that should be conclusion there, gram-positive coccus. And that narrowed it down to these species. The next test the person ran was the catalase test. It was catalase positive. That narrowed it down to these 4 species. The next test the person ran was the PR mannitol test, and there's PR mannitol negative. That narrowed it down to these 3 species. The next test the person ran was the PR glucose test, and the result and conclusion was it was acid. That narrowed it down to these 2 species. They then ran the citrate test, and it was citrate positive. That narrowed it down to this 1 species, Micrococcus varians. So you should have a table or a diagram detailing how you identified your unknown species. And obviously, you have a number which represents one of these species. Don't follow this because you will be following the species here. So it might be a little different. 1, 2, 3, 4, 5. 5 Streptococcus. Let's see what there are here. Where do I have it? Oh, here it is. 2, 3. Yep, see, they only have 4 Streptococcus, and you'll have 5. Any questions about the table? You must include either a table or figure detailing how you identified your species, meaning you give the test which eventually identified your species. Must also give a microorganism paragraph, which is a paragraph to half a page, talking about your organism, including the diseases, the mode of transmission, sources, places it's found, various treatments for that organism, and anything interesting about the organism. So this would be at the end of your report, and you should give a paragraph or 2 or half a page at most on this, and you should give a citation for where you got that information. If you quoted someone, you should give a citation too, an index citation, and then give a reference at the very end of your report. In the very least, you should have where you got your information for the microorganism paragraph. You can use whatever citation style you like. Do not use plagiarism. If you get plagiarism on this project, you will get a 0 for this report. Your report will be graded for plagiarism, meaning it will be plagiarism tested. I will also be testing for AI. Like if you wrote a paragraph and you got it from AI, you should put it in quotes and say where you got that from. Even if it's only a sentence, you should put it in quotes and say where you got it from in an in-text citation, and then give a formal reference for where you got it from. So your report will be checked for plagiarism. It will be checked for AI writing. And then the grading, I've already shown it to you. But here's a section on how the grading will be done. Majority of the project will be the logic of the test you perform. You will be graded something. Let me look that up again. For getting the correct species. Where do I have that? I think I've closed it down. Let me go look it up again. You will be graded 10 points if you get the correct species. If it's the species that's similar, meaning in the same group, you get 7 out of 10. If it's not similar, but the gram stain is correct, you'll get 5 out of 10. If you get the gram stain incorrect, I've only had one student ever do that. They got 0 out of 10. So was the utilization of resources appropriately, meaning we're all test correct. You didn't have an irrelevant test performed. It's 10 out of 10. One test didn't help you at all. When you requested it, you get 8 out of 10. Two tests, 6 out of 10, etc. Was dichotomous logic used? Like I already talked about that one. If dichotomous logic used, you get 10 out of 10. Was all the relevant tests used, meaning you had enough tests to identify the species, no tests were omitted, 10 out of 10. If one test was needed to identify the species, you get 8 out of 10. If two tests were omitted, you get 6 out of 10. Usually only students who wait to the very end and then work on this project will be the ones who have tests omitted. So you should get started on this project right away. It'll take at least 2 to 3 weeks minimum to get this project done if you're following dichotomous logic. So you should get started right away. Do not wait to the end. If you wait to the end, you may not have enough time to get the project done following dichotomous logic. And if you don't follow dichotomous logic, you'll get a penalty, like 3 out of 10. You use all the tests at one request. Were the correct species included at each step? Once you request a test and you get the results, you should, in your unknown notes page, as well as in your unknown report, state which species are under consideration. You don't have to do that at the beginning because all species are under consideration. But once you narrowed it down to like the gram stain, you should state that this narrows it down to the gram positive species. And for the first one, you only need to do that. But after you state the gram stain, the gelatin hydrolysis, and the PR lactose test, you should actually write out the species under consideration. And then you request your next test. You get the test request. You should then write out the species under consideration. And that should be done both in the unknown notes pages as well as in the unknown report. So if you don't put out any species, you'll get over 5 incorrect species. Or if you have over 5 incorrect species, you get 2 out of 10. All species are correct, you get 10 out of 10. If one species at any point in time is incorrect, you get 8 out of 10. 2 to 3 species incorrect, you get 6 out of 10. Completeness of the report, all information is presented, 10 out of 10. One instance of missing info, 8 out of 10. Like if you don't have the table in the document, that'll be 8 out of 10. 2 to 3 instances of missing information, 6 out of 10. Effective, clear communication, meaning is the English, grammar, spelling, punctuation all good? If it's excellent, 15 out of 10. You get a few errors, but the logic is understood, 13 out of 10. Some errors, but logic is still understood, 11 out of 10. Some errors and the logic is not always understood, 7 out of 10. Lots of errors, hard to read. Some errors, I don't understand what you're referring to, 1 out of 15. Now we talked about the species. So for a total of 75, and you'll get the points out of 75. Any question about the grading? Where are we in the project? All right, you should be organized, clear, neatness of the report.
Question.
Uh-huh.
So the table that you showed us, that's the example of the project?
No. About
Do you
an
have an
example of a
example?
project. Let me get to that in just a minute. I'm almost finished with talking about the report, and then I'll show you an example of the project. So each
Oh.
student must submit a report using your own words wherever possible. If not, put it in quotes and give a in-text citation for where you got it from. You do not need to go through this, but in the lab, we would give you a tube that had 2 species in it. We decided for the unknown project when we went online after COVID-19 to just give you a number which represents only 1 species. So you can go through this where you pretend to isolate the species, streaking it out on plates, streaking it out 2 times, and then setting up your stock culture, and then pretending you use your stock culture to set up each of your test requests. And then this is an example using dichotomous logic, and I've got a few cases of dichotomous logic. This is just here in case you need to understand dichotomous logic, and this is going through a dichotomous logic example, and then there's some questions for you to answer. You do not need to turn in these questions for your report. If you don't understand dichotomous logic after going through this example, then speak to me about dichotomous logic. We have another place where I've got an example of dichotomous logic. There's building a dichotomous game plan. You should be using a dichotomous game plan to figure out your project. So this goes through a dichotomous game plan, how to build one, how to use dichotomous logic. I think there's some questions in here too, but going through a dichotomous logic. And here's an example of dichotomous logic. Initially, you have all possible species, A, B, C, and D, and you request a test. In this case, was the species rectangular or not rectangular? Let's go with rectangular. So we cross this out, and then we don't follow these. And so that leaves it to species A and B. They're rectangular. And then you ask, is it not white? Or is it a white species? And if it's not white, it's species A. And if it's a small white species, it's species B. And this is using dichotomous logic to identify the species. Another example would be the table I gave you here. Oh, that's the key. Close that down. Didn't know I changed that. So they initially did the gram stain, separating the species into gram positive and gram negative. Gram negative aren't shown because we're not following those species, so you cross those out. And then you follow these species. The next one that was done was the catalase test, which can be catalase positive or catalase negative. We're not following the catalase negative, but that's actually all of these species here. And the catalase positive were the results. So we're following these 4 species here. The one did the PR mannitol. The test was PR mannitol negative. These are these 3 species here. We're not following the catalase PR mannitol positive, so we're crossing out Staphylococcus aureus. Do you see? That's how we use dichotomous logic. And then you just work until you get 1. And that's Micrococcus varians. We crossed out Staphylococcus epidermidis because it's citrate negative. And our result was citrate positive. And that's using dichotomous logic. Everyone see that? If you're confused on dichotomous logic, just go through I've got 2 examples here. One that's actually in the end of the last page of the document. And then there's one in week 5 lab, building the dichotomous game plan, which I just showed you right here. So that's how you get to your unknown nodes page. You can also get to it from the gradebook. And then you can click here for the unknown grading. This will be where you're putting the document. You will not have a Oops, this is the key, sorry. So I got that way down at the end when you turn it in. The unknown notes pages will close November 28th. So that'll be the last time you can request a test. If you request a test on November 28th, it must be a test that you can get the results in one day because the unknown project is closing November 29th. You must submit your unknown report at 11:59 p.m. or earlier on November 29th. And this is where you submit it. You'll have probably an upload button instead of a speed grader button there. Here, somebody asked about the example. The sample journal is an example of an unknown report. And instead of reading it here, let's go to my online because it's easier to see. So you should have your unknown number, state who it is, state what the class is, the date, obviously. And you should have that on the very top of your unknown report text, meaning I don't want that on a separate page. I used to print these out. I won't anymore, but that was just sort of a waste of paper if you made the cover sheet page. And then you should have the day that you have your first data, like February 3rd. This person streaked out 2 TSA plates, 2 EMB plates, and streaked them out for colony isolation. And then on February 4th, looked at the plates, seeing 2 different colonies on the plates, a white colony and a yellowish colony. Putting photos in your report are helpful, but they're not required. Oops. So this is the observation on February 4th. And then you should have the activities he did on February 4th, where he streaked out colony B on a second TSA plate. I did this to streak it out 2 times to get a pure culture. And then any results he could have, like he could put on here that colony A is yellow in color and small, and what is that, convex? Colony B, which I don't have discussed here, is the white colony, so you could put that in, saying it's a small white colony. You could put in your conclusions. All right. And then on February 5th, he looked at his second streaking, took cells from this colony here to set up his pure culture. Anyways, those are the observations. Oh, he's actually doing this here, where he put in the colony characteristics here. That actually shouldn't be, well, I guess it could be an observation, but if you have any conclusions, you should put those in the conclusion section, which would be after the activities. And then he took colony B here, using this to set up his stock culture, and he's incubating those. And then February 6th, he made observations on the stock culture here. The activities he took from the stock culture to set up the gram stain. You're not quite going to be doing that, but you can pretend to do that. And then the results and conclusions of the gram stain are here. So the cells stain purple and they're spherical cells. That's easy to see there. And then the conclusion is it appears to be gram-positive cocci. So generally speaking, on all days, you should have a observation, an activity. Those are the tests you're going to be running that day starting, and then the results and conclusions. On some days, if you simply request a test and you don't have any results to or conclusions to put up, you'll only have maybe observations from a previous test you requested and the activities you performed on that day. So the results and conclusions will be on another day. For the gram stain, obviously, they're usually coming on the same day. If you did an activity on this day, but you have no observations from a previous test, there will be no observation on that day. And so you might only have activities because if the test takes more than a day, you won't get the results on that day, as well as the conclusions. Any questions about how you write the lab report?
Yeah, so you said you were going to be sending us the first couple, is that right?
The first 3 tests.
Okay, so sh when we write this, should we like write it in a way saying today we streaked out for a certain test or something, or should we just say we requested a test today?
You should state that you streaked out your cells for getting isolated colonies, so you should start that today.
Okay.
And you should also say that you started the gelatin hydrolysis test today, because I'll send that out. You'd need to start this test today so I can send you the results next Tuesday. So this test will be started today. However, the PR lactose test will not start until Monday. So you shouldn't state it today. What would that be? That'd be like 6 days from today. So you should be starting the PR lactose test on 10/27, and I'll send that out to you when you're starting it. And that should be in your report. And then the gram stain will be started on the 7 days from today. So that'll be the 10/28, and you'll get your results on the gram stain on the day you started at. Any question about any of that? It may be helpful to do the gram stain first when you're writing your report so that you narrow down the bacteria first. Like this one put, well, actually, that's not true. He did start with the gram stain because that's not really a testy round. That's just setting up the stock culture. All right. Any questions about the example? The example was actually ran in a real microbiology lab, so it's going to be done a little different than what we are doing, but in a way, you're going to be a little easier because I'm just going to give you one number and it'll represent one species. If you were to come into the real microbiology lab, you get a tube that had 2 species in it, and you have to isolate it out and then take one species, purify it out, and run your tests only on that one species. All right, let's go through. Here's the table that the person did. It's actually an instructor. So he separated them into gram positive. And then he did the pigment, having 3 that were pigmented. Then did the catalase test, in which case they're all catalase positive. So that didn't really help him from the pigment test. He didn't need to put Staphylococcus epidermidis here because it's been excluded by the pigment test, but he's got a different color to say it's excluded. He then did beta hemolysis and got, or he did the hemolysis test and got beta hemolysis. So effectively, he's identified which species it is. And then he ran 3 2 tests after running beta hemolysis, which he would get a penalty in my grade book here because I'm only allowing you to do 1 confirmation test, meaning once you've identified your species, you can only do 1 confirmation test to make sure that you've identified the correct species. And this person has done 2 confirmation tests. And if you were to do that, you get a penalty because you can only get 1 confirmation test. And that's the species he's identified. I'm not sure why he did this one because it looks like he got the same results as Biolesculin. So this test didn't even help him. You don't have to do the confirmation test. You can if you want, but once you've identified the species, you can end the project there. But you are allowed to do 1 confirmation test. And let's say you're not totally confident about the color, and so you're questioning whether is it really these 3 or might it be, I don't know, Staphylococcus epidermidis. And the catalase test was positive for Staphylococcus epidermidis. And so you could do 1 confirmation test to, I guess, rule out Staphylococcus epidermidis, which this one actually would because Staphylococcus epidermidis is not Biolesculin negative. I think I forgot to mention that when discussing the project. You can do 1 confirmation test. All right, any questions about this project? I think I'm done here. Oh, let's talk about the example. Paragraph about the species. You don't need to do bullet point. You can do text. It's fine. You should give an in-text citation here where we got that information. He's not doing that. You should give an in-text citation and then the references, which are the full text reference. He's got 2 of them. Staphylococcus aureus in a healthcare setting from the CDC, and then he's using the textbook as well. 6th edition. Boy, that tells you how long ago this was down there, like on about 13th edition now. Pearson Publishing, New York. I think the author has changed too. I think there's more than 1 author. Oh, wait a minute. Bauman. Oh, wow. We don't even use Bauman anymore. We use Tutora. I didn't know they used Bauman at Clark College just because this is so long ago, before I was working here. All right. Any questions about the project? I didn't change it, did I? I think that's it. So, you'll have to wait until I send out the email with your number. Although you could write up on the unknown notes page that you're streaking out the cells that I gave you to get isolated colonies, and then you'll do it a second time to get a pair culture. Any question about any of that? Any questions at all? All right, that's it. Say goodbye. Bye.